Welcome to video lecture of Biochemistry. I am Dr.Merina, Associate Professor of Department of Industrial Biotechnology, Faculty of Engineering, Bharat Institute of Higher Education and Research. So today in Biochemistry, we are going to discuss about structure of protein. So in this topic, we are going to discuss about the general introduction and amino acids, formation of peptide, structure of protein, and summary of structural level of proteins. So proteins are the most abundant organic molecules of the living system. We know that the organic molecules means which will be having the basic atoms like carbon, hydrogen and oxygen. And they constitute about 50% of the cellular dry weight. They constitute the fundamental basis of structural and functions of life. So in 1839, Dutch chemist G. Mulder was the first to describe about the proteins. So the term protein is derived from a Greek word that is called proteos. This is the meaning first place. So the proteins are the nitrogenous macromolecules that are composed of many amino acids. So they are the polymers. Actually, we can say that they are the biopolymers of amino acids, and they made up of one or more polypeptide chains. So every protein in its native state has a unique three-dimensional structure, which is referred to as its conformation. So the number of and the sequence of this amino acid in the proteins are the different in different proteins, and because of this sequence and the numbers, they'll be defining their functions. So the functions of a protein arises from its conformation. So before going to the structure of the different level of proteins, we should know what is the configuration and what is the conformation of a molecule. So when you're talking about the configuration of the compound, that denotes the spatial relationship between the particular atoms. Like example, you can say that L amino acid or D amino acid. We know that in case of the L amino acid, where the NH2 group, amine group will be there in the left hand side of the alpha carbon. And in case of the D amino acid, that alpha carbon, that right hand side will be the amino acid. But when you're talking about the conformation of molecule, this means the spatial relationship of every atom in a molecule, like rotation of a portion of the molecule. So now we'll be talking about the amino acid, because we know that the proteins are biopolymers and they are made of monomers. So the individual monomers are called as amino acids. So amino acids are a group of organic compound. So they are also made of what? Carbon, hydrogen and oxygen. As a name, it is designating that amino acid. So amino means it is a functional group. Acid means it is another functional group. So it is containing two functional groups in the same compound. One is amino group, one is the carboxyl group. So we know that the amino group, the NH2 group is the basic, while the carboxyl group, the COH, it is in acidic in nature. So in nature, we can find out around 300 amino acids, but out of that, only 20 amino acids only will be utilized for the protein synthesis. So now we see the structure of amino acid. So there are carbon will be having the 4 valency. That means 4 hands will be there. So this carbon is called as alpha carbon. Why? Now in this carbon, there will be 2 functional group will be attached. So in the same carbon it is attached, that's what it is called that alpha carbon. So in this carbon, in one hand will be their amino group, NH2 group, another hand will be their COH group, another hand will be their hydrogen, and the rest hand will be their side chain R. So this, just now I told you that there will be 20 amino acids. So there will be 20 R's. So based on the 20 R's, there will be 20 amino acids. So now here we are given categorization of the amino acid. So basically mainly 5 groups will be there. So that non-polar aliphatic R groups. So in that there will be 6 amino acids: glycine, alanine, valine, leucine, methionine and isoleucine. So that functional group, if you see that there will be simple to the complex one. So in the glycine there will be H. And if you see the isoleucine, there will be CH, CH3, CH2, CH2. So it will be more simple to more complex. Next group will be coming polar uncharged R group. In this also there will be 6 amino acids. That will be serine, threonine, cysteine, proline, asparagine and glutamine. Next category will come positively charged R group. So in this will be there 3 amino acid only. There will be lysine, arginine and histidine. They are also called as a basic amino acid. Next will be coming acidic amino acid or negatively charged R groups, that is aspartate and glutamine. And last group, that is a non-polar aromatic R groups. In this, there will be phenylalanine, tyrosine, and tryptophan. Among this, the tyrosine also will be calling as a hydroxyl amino acid because it will be having the OH group in this benzene chain. So if you see that the positively charged R group, in this we will be having the 3 amino acids like lysine, arginine, and histidine. So they are called as a basic amino acid because apart from the that general NH2 group, it will be having an additional NH2 group. So that's what it will be giving the basic nature for this amino acid. Where in case of the acidic amino acid, instead of the common the CO minus group, additional CO minus will be there. So that's what it will be calling as a acidic amino acid. So the classification of amino acid, if we see that 4 category, non-polar, that means that is a hydrophobic in character. In that will be there alanine, valine, leucine, isoleucine, proline, phenylalanine, tryptophan, and methionine. And in case of the polar group, it will be in hydrophilic. Okay, so they will be glycine, serine, threonine, cysteine, tyrosine, asparagine, and glutamine. Next will come the acidic, that is a negative charge that we have seen that aspartic acid and glutamic acid. And in case of the basic group, that will be positively charged lysine, arginine, and histidine. So now amino acids are over. So next one we will go to the protein structure. First we have to formation of the peptides. So when the 2 amino acids will be joining, we will be getting peptide. So first we have to learn what is the formation of the peptide bond. So it is a peptide bond is a type of covalent bond and it is formed by the amide linkage. Amide linkage means CONH. So it is a CONH linkage between the alpha carboxyl group of one amino acid and the alpha amino group of next amino acid. Okay, and when this that COH and the NH2 group will be joining together and they'll form a amide bond, that time one molecule of water will be released. Okay. So the successive amino acids are joined or cemented, we can tell, by the peptide bond, that's the CONH in the protein. So this is the basic bond for any protein structure. CTAs will be having 40% double bond character, which will be caused by the resonance. And peptide bond forms strong backbone of the polypeptide and side chains of amino acid that will be project outside the peptide backbone. So how it will form? So if you see this, the alpha carboxyl group of the one amino group with the side chain R1. Here we have taken 2 different R1 and R2. It may be same also. That is also possible. So they'll form a covalent peptide bond with the alpha amino group, the another amino acid with side chain R2 by removal of one molecule of water. So the dipeptide will be formed. So the dipeptide means it is not the 2 peptide bond. It is 2 amino acids are joined. So dipeptide means 2 amino acids joined, but what? Single peptide bond. So when we'll be talking about the tripeptide, it means 3 amino acid and 2 peptide bonds. So the dipeptide can then form a second peptide with another third amino acid, that is maybe a side chain R3 to give tripeptide. So the repetition of this process will generate a polypeptide or protein of specific amino acid sequence. So now we have to learn what is the characteristics of a peptide. So it will be rigid, covalent, stable and strong and can be hydrolyzed by the action of the proteolytic enzymes, maybe acid and alkalis. So it will be in planar in nature and partial double bond character because there will be no freedom of rotation and it will be having transposition. So like CO and NH, they will be in like just opposite. That's what it will make this structure is more stable. Okay, so the both groups will be polar and this will be involved in the hydrogen bond formation. And this, the hydrogen bond formation, we can see in the later in the protein different level of structure, it will give the additional stability for a protein structure. So impart from the stability to the primary structure of the protein, disulfide bonds also will be responsible for the stability. So the side chains are free to rotate on either side of the peptide bond and distance between the amino acids is 1.32 angstrom, which is the midway between that of the single bond. That will be around 1.49 angstrom and the double bond around 1.27 angstrom. So in case of the polypeptide chain, at one end there will be one free alpha amino group, that is alpha N terminal end, and the protein biosynthesis starts from the amino terminal end. That will be the first amino acids. The other end of the polypeptide chain is carboxyl terminal end. So that is called the last amino acid, where there is a free alpha carboxyl group. So conventionally, when you'll be taking a protein, when you want to depict its name, amino acid sequence, we have to start with the N terminal end and we have to finish with the C terminal end. So N terminal amino acid and C-terminal amino acid. So all other alpha amino acid and alpha carboxyl groups are involved in the peptide bond formation. So when we'll be writing up the peptide structure, the amino acid sequence is written from the left to right. With the free amino acid, that is called N-terminal amino acid or residue, number 1 by tradition on the left hand side, and ending with the free carboxyl again, C-terminal amino acid residue. So the shorthand to read peptides, how will be designating them? So there will be 3-letter or 1-letter abbreviation for all the amino acid, and the shorthand form of the amino acid in protein to be read from the N-terminal residue on the left hand side of the peptide. Like suppose if you having the glycine, alanine and valine, so we'll be having GAV. So G means it denotes that G will be attached with the NH2 group and V is attached with the COH group. So naming of the peptide, how will take? For the naming of the peptide, the amino acid suffix in, like glycine, whoever it is there, it will be glycylide. YL will be finished. And the tryptophan or et, okay, on AN or et, like glutamate. These are all will be finishing with YL with the exception of C-terminal amino acid. Why no? Like C-terminal acid will be in the right hand side. So we no need to extend this name. So now here we are given the polypeptide chain, which will be showing the N-terminal N and the C-terminal schematic diagram. So N-terminal N, it will be finished with an NH3 plus and C-terminal will be finished with the CO minus. So all the R1, R2, R3, R4, R5, whatever may be the number, it will be attached with the carbon. Okay, and in between there will be CONH, that is called the amide bond. So the polypeptide backbone is the repeating sequence of the N, CCNCC in the peptide bond. So the side chain of the R group is not the part of the backbone or the peptide bond. So they will be the side chain, they will be extending. This R will be giving extra additional support or stability for the protein structure. So now we'll come. So amino acids we learned the peptides. So now we'll be learning the protein structure. So there are different level of organization of protein structure. So biochemists have distinguished several levels of structure of organization of protein. So protein structure can be classified into the 4 levels of organization. They are primary structure, that is amino acid sequence, secondary structure, that is a regular substructure, then tertiary structure, that is a 3-dimensional structure, and the quaternary structure, that is called complex of protein molecules. So once the complexity will increase from the primary structure to the secondary to the tertiary to the quaternary, and that will give the protein a proper function. So the 4 orders of structure, if you see that primary structure, secondary structure, tertiary structure and quaternary structure. So quaternary structure, we can see that it is an assembled subunits. Subunit nothing but a polypeptide chains. So that will be discussing later. So in general, proteins are composed of L-alpha amino acid. So I told you that in case of the L-alpha amino acid means where the NH2 group of the basic amino acid structure will be in the left hand side. And the structure of the proteins is rather complex, which can be divided into this 4 level of organization. So the primary structure, if you see that this is just on the assembly of amino acid by peptide bonds, normal covalent linkage, amide linkage. When you come for the secondary structure, the same that polypeptide chain, the single polypeptide chain, it will be some sort of folding will be there. So it may be alpha helix or pleated sheet will be there. Next, the complexity will come to the tertiary structure. Here we can see the packing of this secondary structure. Okay, so that is called the 3-dimensional structure will form, and last is the quaternary structure. So similar type of that 3D structure of the one polypeptide chain, there will be more subunits will come and join. So there will be interchain interaction, and because of that we'll be getting quaternary structure. So the 4 level of structural organization just will give the definition of this. So the primary structure, which is determined by the number and the sequence of amino acid in the protein. Where in the secondary structures, we can see that it is the conformation of the polypeptide chain, which will form by the twisting or folding. So 2 things will be there. So it occurs when the amino acids are linked by the hydrogen bonds to form the alpha helix and beta sheet. So now I told you that there will be backbone will be the peptide, but the side chain will be the R group. So the R group will be forming this hydrogen. So now we'll talk about the tertiary structure. It is a three-dimensional arrangement of the protein structures. It is formed when the alpha helices and the beta sheets are held together by weak interaction. So there will be having the additional bonds and all. Next we'll come to the quaternary structures. It occurs in the protein that is called the oligomers, consisting of more than one polypeptide chain, where the certain polypeptide aggregates to form a functional protein. So here we can see that individual structures. So the primary structures will be determined by the sequence of the amino acid. But the secondary structure, it is a number and the sequence amino acid in the protein, and this will be occurred by the hydrogen bonds. So there will be mainly two types. One will be the alpha helix, another one is a beta pleated sheets. Next will come to the tertiary structure, which will be the three-dimensional arrangement of the protein structure. And the last one is a more complex structure, that is a quaternary structure. This occurs in the protein consisting of more than polypeptide chain, where the certain polypeptide aggregate to form the functional protein. So what are the forces involved in the protein structure? So mainly in case of the primary structure, we have seen that the peptide bond. But in case of the secondary, tertiary and quaternary structure, apart from additional of this covalent bond, there will be some weak forces. Okay. So now first we'll explain the primary structure. So the primary structure of the protein denotes a number and sequence of amino acid in protein. So the successive amino acids are linked by the peptide bond. We know that it is a type of covalent bond, and generally amino acids are arranged as a linear chain. So the each component of the amino acid in a polypeptide is called as a residue or moiety. So the primary structure of the protein is largely responsible for its function. The branching point in the polypeptide chain may be produced by the interchain disulfide bridges, the covalent disulfide bonds between the different polypeptide chain in the same protein, or portion of the same polypeptide chain, that is called the intrachain. So they are the part of the primary structure. So each polypeptide chain will have an amino terminal, that is called N-terminal, that will be the left hand side in the free amino group, and a carboxyl terminal ends, that is the C-terminal with a free carboxyl group, which will be in the right hand side. So by convention, they are represented with amino terminal on the left and the carboxyl terminal on the right hand side. So the amino acid composition of the protein determines its physical and chemical properties. So the primary structure of the protein, that is called sequence of amino acid, is determined by the gene contained in DNA. So the primary structure of number of proteins are known today, like insulin, glucagon, ribonuclease, growth hormone. So these are all we can prepare in biotechnology with the help of the rDNA technology. So the any change in the primary structure of the protein, it will affect their functions. So what is the clinical applications of the primary structure? So the presence of specific amino acid at a specific position or in number is very important for a particular function of a protein. So even one single amino acid change, not even change like changing of the position, changing of the sequence, it may change its function. So any change in the sequence, it will make the abnormality and may affect the functions and properties of this protein. And the many genetic diseases result from the protein with abnormal amino acid sequence. If the primary structure of the normal and mutated proteins are known, that this formation may be used to diagnosis for clinical study of this disease. Next we'll come to the secondary structure. So the three-dimensional confluence of the polypeptide chain may be by twisting or folding is referred to as the secondary structure. So it is a local, regularly occurring structure in protein and is mainly formed through hydrogen bonds between the backbone atoms. So the Pauling Corey and Corey, this is the two scientists, they studied the secondary structure and proposed two conformation, that is the alpha helix and beta sheets. So secondary structures are the special arrangement of the protein by twisting and of polypeptide chain or folding of helical coiling patterns in protein, that is called alpha helix, or zigzag linear, that is called the beta sheets. Or sometimes we can see the mix formed by the hydrogen bonding and disulfide bonds. So the secondary structures denotes the steric relationship of the amino acids close to each other. One of the form of the coiling in the polypeptide chain is right-handed alpha helix. So since the proteins are made up of alpha amino acid, the coiling of polypeptide chain into the right-handed alpha helix is mostly we can form. So thus there will be super secondary structures also, which will be ending folding patterns in the protein, and that Linus Pauling and the Nobel he got 1954, and Robert Corey, that is he got a Nobel Prize at 1951, proposed the alpha helix and beta pleated sheet structure of polypeptide chains. So first we'll see the alpha helix. So it is a right-handed spiral structure because of it is made of all L-alpha amino acids, and these side chains are extended outwards. And it will be stabilized by hydrogen bonds that are arranged such that the peptide carboxyl oxygen and the amide hydrogen, they can make the hydrogen bond. So amino acids per turn will be around 3.6 and pitch is around 5.4 angstrom. So the alpha helical segments are found in many globular proteins like myoglobin, troponin C, etc. So length will be around 12 residues, and there will be 3 will be in helical turns. So there will be phi angle around minus 60 degree and the psi angle will be around 45 degree, and this falls within the fully allowed regions in the Ramachandran diagram. So the characteristics of alpha helix, if we see, it is the most stable conformation formed spontaneously with the lowest energy. So right or left hand is spiral helical or tightly curled structure in the stable form. A right-handed helix turns in the direction that the fingers of the right hand curl when it is thumb point in the direction of the helix rises. So it will be stabilized by hydrogen bonds like we know that the hydrogen bonds, they are weak and it is strong enough due to the large number of stabilizer alpha helix structure. And these hydrogen bonds are the main chain which is in the form in the backbone, and side chains of the amino acid extend outward from the central axis. Hydrogen bonding occurs between the carboxyl oxygen of one peptide and the amide nitrogen of another peptide bond, and 3 amino acid residues will be up or down in the chain. And like we can see that all the peptide bonds except the first and the last polypeptide chain, it will participate in hydrogen bonding. So as the number of hydrogen bondings are more, so this will give the more stability to the alpha helix structure. So each peptide bond in the polypeptide chain participate in the intra-chain hydrogen bonding. So it will be a single polypeptide chain. So each turn of the alpha helix will be containing 3.6 amino acid per residue or per turn of the helix with the R group protruding outward radially. So a rise along with the central axis of 1.5 angstrom per residue, and it travels distance around 5.4 nanometer per turn. So the spacing of each amino acid residue, like for axial distance between the amino acid, will be around 0.45 nanometer or 1.5 angstrom translation. So the right-handed alpha helix more stable and common than the left-handed helix, and left-handed helix are rare because of the presence of alpha amino acid found in the protein, which excluded left-handedness. So the proline, hydroxyl proline, and glycine, the 3 amino acids, they will be disrupting alpha helix formation, and this will introduce a kink or a bend in the helix. So if you want to break the structure, we can introduce this amino acid to the alpha helix. So the large number of acidic group like aspartic acid or glutamic acid and the basic amino acid like lysine, arginine, histidine or amino acid with the bulky R group disrupt the alpha helix. So abundant in myoglobin, hemoglobin, keratin of here, that's what it is called as alpha keratin, and proteins in wool, it is found. So here we have shown the right-handed and left-handed alpha helix structure. So right-handed alpha helix turns in direction that fingers of right hand curl when thumb points in direction of helix rises. And left-handed, that is alpha helix turns in direction that fingers of left hand curl when the thumb proteins in direction of helix rises. So this is the formation of the hydrogen bond in the alpha helix. So you can see that R1, that CONH will be making the bond between the R4 and R5, that CONH, and that nitrogen oxygen distance will be around 2.8 angstrom. So helix stabilizing amino acid, we know that helix destabilizing, that means helix breaker, we can say glycine and proline. And why proline as a helix breaker? Since nitrogen of proline residue in a peptide linkage has no substituted hydrogen, okay, as it has an imino NH group instead of amino group. And for this formation of the hydrogen bond with other residue, proline first fits only in the first turn of an alpha helix. Otherwise, it will bend the term. And glycine, how it will be, like all bends of alpha helix are not caused by proline residue, but bend of 10 occurs, often occurs at a glycine residue as side chain of glycine is small. So what is the structural importance of alpha helix? So several alpha helices can coil around one another like a twisted twinned cable, forming a strong stiff bundles of fibers and give the mechanical support. Next, we'll talk about the beta plated sheets. So this is the second structure of the secondary structure. It will be formed when the 2 or more polypeptide line up side by side. So there will be individual polypeptide that is called the beta strand, and each beta strand is fully extended. They are stabilized by hydrogen bond between NH and carboxyl group of adjacent chain. So what is the characteristics of beta pleated sheets? So it is a beta because it was second type of structure after the alpha helix as elucidated. So when the alpha helix keratin is stretched, the hydrogen bonds are broken and new hydrogen bonds are formed between the CO and NH of the adjacent parallel chain or neighboring molecule. So this is called beta pleated sheets. So it is also stabilized by the hydrogen bond, and they are composed of two or more segments of fully extended polypeptide chain. Now this hydrogen bond occurs between the two polypeptide chain, that is called hydrogen bonds are intra-chain, or two region, that means neighboring segments of a single chain of polypeptide chain, that is called hydrogen bonds are inter-chain. So there are two type of beta sheets we can find, two varieties, that is called anti-parallel beta sheets, where the neighboring hydrogen bonds polypeptide chains are in opposite direction. And one will be the parallel beta sheet. So there will be hydrogen bond chains extended in the same direction. So in case of the parallel, that in both chain in will be in the same side and the C will be the opposite side. But in case of the anti-parallel, the N and C will be there for the chain. So that's what, by that we can, the direction we can say that it is a anti-parallel. So what are the characteristics of beta pleated sheet? So there will be two polypeptide chain in a beta pleated sheet may run in the same direction, that is called parallel, with regard to amino acid and carboxyl terminal ends of the polypeptide chain, or in the opposite direction, that is called anti-parallel beta pleated sheets. So the major distances between the adjacent amino acid residue will be around 3.5 angstrom, and a major structural motif in the fibrin of silk, like anti-parallel you can see, flavodoxin you can see the parallel, carbonic anhydrous, that is the one enzyme, in that both like anti-parallel as well as the parallel we can see. Some regions of globular protein like chymotrypsin ribonuclease also you can see these beta pleated sheets. So the two major sorts of connection and between the beta strands are a hairpin structure and a crossover structure. So hairpin or same and connection, same end connection, and where the crossover it is called the opposite end connection. So in case of that crossover also, you can see that two types. One is a right-handed crossover, one is a left-handed crossover. So what is the clinical application of the beta pleated sheet? It is a secondary structure of the protein which is found in the both fibrous and globular protein, and the anti-parallel beta sheet conformation is less common in human protein. So we can see the where we can found this beta sheet. So silk fibrin, it is the best example of the beta pleated sheet, and apart from that amyloid in human tissue. So we know that it is a protein that accumulates in amyloidosis and or Alzheimer's disease. This is called dementia occurring in the middle age associated with this amyloidosis. So now we can see that polypeptide chain conformation, how it will be there. So the backbone of the main chain of a protein refers to the atoms that participate in peptide bond, ignoring the side chain of the amino acid. So they only the reasonable free movement or rotation around the C alpha and N bond that will be measured as a phi, and the bond will be measured as a chi. So these angles both are defined as 180 degree when the polypeptide chain is in full conformation. So the conformation of the backbone can be therefore described by the torsion angles. So this is also called as the dihedral angles or the rotational angles. So based on that, we can find out the type of secondary structure. So this will be explained by the Ramachandran plot. This Ramachandran plot will be able to help to understand the backbone of amino acid residues. So the amino acids with the larger side chain will show the less number of allowed region within the Ramachandran plot. So we can see that in this Ramachandran plot, it will be plotted by psi versus phi, and we can see that beta sheet, the region, left-handed alpha helix as well as the right-handed alpha helix. So the Ramachandran plot is a way to visualize the backbone dihedral angles like phi against the chi of the amino acid residue in the protein structure. So a Ramachandran plot can be used to show which values or conformation of the phi and chi angles are possible for an amino acid residue in a protein, and to show the empirical distribution of the data point observed in a single structure. So each region will be designating a particular conformation of the protein. So the darkest areas correspond to the core region that will be representing the most favorable combination of the phi and chi values. Next, we'll come to the protein folding. So once we'll go for the like more complex structure, the peptide bond allows for rotation around it, and therefore the protein can fold and orient the R group in favorable position. That's what it will make the protein in a sturdy structure. So the weak non-covalent interaction will hold the protein in its functional shape. These are the weak and will take many to hold the shape. So protein folding occurs in the cytoplasm. So the two regular folding patterns have been identified that form between the bonds of the peptide backbone. So one is the alpha helix, that is a protein turns like a spiral fibrous protein like hair, nail or horns we can see. And one is a beta sheet, which will be protein folds back on itself as in a ribbon, or that we can find in the globular structure. So now we'll call about the turn or bend. So in addition of the alpha helices and the beta strands, a folded polypeptide chain contains two other types of secondary structure, that is called loops and turns. So loops and turns connect alpha helices and beta strands. The most common type cause a change in direction in the polypeptide chain and allowing it to fold back to itself to create a more compact structure. So loops have hydrophilic residue and they are found on the surface of the protein, where the loops only will be having 4 to 5 amino acid residue, and they are called turns when they have internal hydrogen bonds. So reverse turns also are there. They are form of the tight turn where the polypeptide chain make a 180-degree change in its direction. So reverse turns are also called beta turn because they are usually connected adjacent beta strands in a beta sheets. So now we can see that the beta turns, this is also called as a beta bends or tight turns. So it permits the change of direction of the polypeptide chain to get a folded structure. It gives a protein globularity rather than the linearity because it is a more compact. So in a beta turn, a tight loop is formed when the carboxyl oxygen of one residue forms a hydrogen bond with the amide proton of an amino acid and 3 residue down the chain. And this hydrogen bond stabilizes the beta bend structure. Proline and glycine, we know that they are frequently found in the beta turns, and proline, because it's a cyclic structure, is ideally suited for the beta turn. And glycine also because with the smallest side chain of all the amino acid, it is the most sterically flexible. A beta turn is a means by which the protein can reverse the direction of its peptide chain. So it can bend actually. So the beta turns often promote the formation of antiparallel beta sheets, and hydrogen bond stabilizes this beta bend structure. And beta turns often promote the formation of antiparallel beta sheets, and also it will be finding in the protein surfaces. So what is the characteristics of beta bend? It is consists of 4 successive amino acid residue and frequently contains the proline and glycine for the both because of the bending, and it is stabilized by intra-chain disciple bridges and hydrogen bonds. It occurs primarily of the protein surfaces and impart globular shape rather than the linearity to the protein, and it will promote the formation of the antiparallel beta pleated sheets. And importance of these beta bends, they help in the formation of compact globular structure. So example is collagen triple helix, we can see, or the silk fibrin beta sheets. So this is the alpha helix and this is the beta beta sheets of the secondary structure. So now we'll be moving for the tertiary structure. Tertiary structure is the 3-dimensional configuration of a polypeptide. So tertiary structures is based on the various types of interactions between the side chains of the polypeptide chain, and the common feature of the protein tertiary structure reveals much about the biological function of the protein and their evolutionary origins. So the function of a protein depends on its tertiary structure. If it is disrupted, the protein will lose its activity. So in this context, we have to discuss about the domains. Polypeptide chain containing more than 200 residues usually fold into 2 or more globular clusters. They are called domains. So fundamental functions and the 3-dimensional structure of proteins, these domains of 10 of a specific function such as the binding of a small molecule. So in this, they will make a cleft where the molecule can bind. That will be most common we can find in the enzymes where the substrate can bind and the catalysis will be done. So the many domains are structurally independent units that have the characteristics of small globular proteins. So what are the factors which will be stabilizing the tertiary structure? In case of the globular protein, tertiary interactions are frequently stabilized by sequestration of the hydrophobic amino acid residue in the protein core. Consequent enrichment of charge of hydrophilic residues on the proteins, that will be water exposed surface. In case of the secreted protein, we can see that disulfide bonds between the cysteine residue help to maintain the protein tertiary structures. So what are the interactions which will stabilizing the tertiary structure? So there will be mainly 5. One is the disulfide bonds, hydrophobic interactions, hydrogen bonds, ionic interactions, and van der Waals forces. So the tertiary structure refer to the three-dimensional conformation of the protein, which will be generated and maintained by the weak forces, that is called valence forces or non-covalent interaction, such as hydrogen bonds. It will form between the CONH of two different peptide bonds or OH group of hydroxyl amino acid like serine and COH group of acidic amino acid like aspartic or glutamic acid. Next, we'll talk about the ionic bonds or electrostatic interaction, or it may call as a Salt bridges also. It is formed between the oppositely charged group. They are in close vicinity. So they are also formed by oppositely charged R group of polar amino acid, like a basic amino acid will be making electrostatic interaction with the acidic amino acid. Next, hydrophilic interaction. This will be water-loving groups of associated with water. Hydrophobic interaction, it will be formed between the hydrophobic groups like aliphatic side chain amino acid like alanine, glycine, phenylalanine. Next will be the van der Waals forces. This will be weak, but collectively it will contribute maximum towards the protein structure. So that during the folding of the globular protein, like it may be a spherical round, hydrophobic groups prefer to the interior and hydrophilic group prefer to be the outside the surface of the protein. So the tertiary structure acquired by a native protein is always thermodynamically more stable. So this is the covalent bond between the sulfur atoms. So two cysteine amino acid, they'll be forming the disulfide linkage. So two sulfur group will be attached. So that's what it is called. It will be disulfide bond. So this is the hydrogen bonds, which will be weak, allows to the broken and reform easily, and it will allow the structural changes and producing functional molecules. This is the electrostatic interaction, where the ions on R groups form Salt bridges or electrostatic interactions through ionic bonds. NH3 plus and CO minus areas of the protein attract and forming the ionic bonds. So next one, the hydrophobic forces. It will be close attraction of non-polar R group through dispersion forces. They are non-attractive interactions, but results from the inability of water to form the hydrogen bonds with certain chains. So now very weak but collective interactions over the large area stabilize the structure. It will repel polar and large molecule particles. Next, we'll talk about the quaternary structure. So the biological functions of some molecules is determined by multiple polypeptide chain or multicritic proteins. So arrangement of this polypeptide subunit is called quaternary structure. So the quaternary protein structure involves the clustering of several individual peptide or protein chains into a final specific shape. So the variety of bonding interactions, including hydrogen bonding, Salt bridges, disulfide bond, they only hold this particular geometry. So there will be two kinds of quaternary structures. Both are multi-subunit protein like homodimer. That will be association will be identical polypeptide chain, or it may be a heterodimer, that will be interaction between subunits of very different structures. So subunit are they'll be holding together by non-covalent interactions. So quaternary structure adds stability by decreasing the structure by volume ratio of smaller subunit. This will be simplifying the construction of large complexes and like viral capsid or proteosomes. So what is the importance of the quaternary structure of the globular proteins? These are the subunits of oligomeric protein, may either function independently of each other or may work cooperatively as in hemoglobin, where the binding of the oxygen to one subunit of tetramer increases the affinity to the other subunit for oxygen. Oligomeric proteins are regulators of cell metabolism and the cellular functions. So if you see the summary of the structure level, you can see that primary, it is a sequence of amino acid. Secondary means the coiled alpha helix, beta peptide sheet, or a triple helix formed by the hydrogen bonding between the peptide bond along with the chain. And the tertiary structure, where you can see the folding of the protein into a compact three-dimensional structure, which will be stabilized by interaction between the side R group or amino acid. Where in case of the quaternary structure, a combination of the two or more proteins are needed to form a larger biologically